and form archisporial cell. So what is archisporial cell? At initial cell or at initial stage, at each side of the corner there are one hypodermal cell become meristematic, and it is called archisporial cell. The archisporial cell by periclinial division give rise to outer primary parietal cell and inner sporogenous cell. Primary parietal cell form the multi-layered microsporangial wall, while sporogenous cell form the microspore mother cell. In our next slide, we are going to discuss what are the parts of primary parietal cell and the sporogenous cell part. The primary parietal cell form the anther wall, and the anther wall consists of a mature anther wall consists of 4 layers. So primary parietal cell helps in the formation of anther wall, and it is divided into 4 layers. What are those? Epidermis, endothecium, middle layer, and tapetum layer. This tapetum layer is the innermost layer, and it is of 2 types. One is amoeboid type and other is secondary type. Amoeboid type means it can form projection and can penetrate into the sporogenous tissue to provide nutrition. And secondary secretory or glandular type, they form glands and some kinds of secretion that helps in the development of spore inside the microsporangium. So anther wall derived from primary parietal cell, and it is differentiated into 4 Ds layer, and the innermost layer is called tapetum. Next is the sporogenous tissue. Sporogenous tissue is developed into a number of diploid mother cell. Those are called microspore mother cell. So here is given the primary parietal sporogenous layer give rise to microspore mother cell. The microspore mother cell, shortly called MMC, are always diploid or twice N. The sporogenous cell in normal way divides several times mitotically and before functioning as pollen mother cell. So at initial stage, the microspore mother cell or sporogenous tissue divided by mitosis to form a number of diploid cells. These are called microspore mother cell. Each microspore mother cell by mitotic division give rise to 4 microspores or pollen grain. Microspores are always haploid, or the chromosome number is always N. So here is the process. You see, this is the initial structure of anther, and at each corner one of the cell become meristematic. Here is the anther during the growth, it shows the elongation considerably, and at each corner they form the archisporial cell. Here is the mature stage. So in our next slide we see here how the microspore are formed. Here you see the 2 lobes of anther. At each corner archisporial cell become meristematic, and it divides into 2 cells, that is called primary parietal cell and sporogenous cell. In our next slide, here also we see single sporangium. Here you see one archisporial cell at each corner. It divided into primary parietal cell and sporogenous cell, and the sporogenous cell by mitotic division differentiate into a number of microspore mother cell, and the primary parietal cell develop into 4 wall layers. The mother cell by meiotic division form 4 tetrads or spore tetrads. That means here 4 spores are found together, and later they differentiated into single microspores. So this is the process of microsporogenesis. In the next slide we see that here is the initial stage. Number A diagram shows the initial stage of the anther before it differentiated into 4 lobe structure. In the B diagram it slightly developed further, and at each corner they form a single meristematic or dividing cell, that is called archisporial cell. And this cell always develop from hypodermal cell, that is below epidermis. In the next slide you see it going to differentiate it into 4 lobed structure, and here the archisporial cell is formed. Here the archisporial cell divide periclinally into inner sporogenous cell and outer primary parietal cell. The primary parietal cell form the wall layer of microsporangium, and the inner sporogenous cell differentiated into a number of microspore mother cell that divided by meiosis into 4 pollen grains or spore tetras. Here it is the mature structure of anther. Here 2 lobes or sporangia are found. Here 1 microsporangium, it is another microsporangium, here also another microsporangium, and here also another. So 4 pollen sacs or microsporangium are found where microspore formation occurs. So this is the structure. In our next slide, here this is the last slide. Here after the formation of microspore, this is the microspore or formation. This is the microspore tetrad. First the microspores are attached together, but later they differentiated into single pollen grain or microspores. Each microspore pollen grain consists of 2 wall layers. Outer is called exine, inner is called intine, and they possess vacuoles also and a large nuclei. And this is the whole structure of pollen grain before germination or pollination. Up to this, it is the process of microsporogenesis. In the later part, we discuss about microgametogenesis after the understanding of this part. So, it is now clear, I think, what is microsporogenesis and how it occurs. Microsporogenesis is the process of formation of microspore is called microsporogenesis. And how it is formed? At each corner of the anther, one of the hypodermal cell become meristematic and it form the archisporial cell. So at each corner one cell become meristematic to form the microsporangium. So how many microsporangia are found at each corner? 1 microsporangia. So in an anther 4 microsporangia are formed, and inside which microsporogenesis occurs. So this is the portion we have discussed today. If you have any doubt, you can discuss with me in the later session. Okay, student, thank you.